So now that we've talked a little bit about absorption of light, I also just wanted to show you an example of what a spectrometer might look like in practice. So this is actually from a few years ago. This is what the spectrometer looked like downstairs in my infrared spectrometer and Hale lab. And we have all these elements that we talked about. I use a light source of a laser. So that's down here. This is what's known as a quantum cascade laser. It produces light. We have a reference cell here that is for calibrating the wavelength of our sample. That's not strictly necessary for other types of spectrometers. Depends on how exactly yours is set up. I also don't have a monochromator, and we'll get to this when we talk about light sources. But lasers sort of have a built-in monochromator. They produce light at a very specific wavelength. So the light gets produced from my quantum cascade laser and then gets sent through. Over here is where our sample is. So this is what's called a multipass cell, and the light bounces back and forth a bunch of times. Now the light isn't red like this. I've just drawn these arrows on. This is infrared light, so it's actually not visible. And so what we do in our, and then the light comes out and goes onto a detector, which is this blue box over here. And so when we're actually taking absorption measurements, what we do is we fill this with nitrogen, and that is our blank that we use, measure the amount of light that makes it onto our detector, and then we repeat the measurement, putting in the sample, in this case isoprene, that we're trying to measure, and we do a comparison between the two. Now, what does this data actually look like? So if we go to the next slide, this is what we looks like if we measure just pure nitrogen. So we get some amount of signal on our detector. Our detector is something that produces an electrical signal in proportion to the amount of light that goes on it. So this is not calibrated. This is just some data from a few years back. But we get nitrogen. My laser doesn't produce a very steady amount of power as you change the frequency of the light. So we see these wiggles. That's just the laser power changing as we change the frequency. So we measure P naught. This is if we have just pure nitrogen as our blank. And then we repeat the measurement with our analyte in there. In this case, we're looking at isoprene. So we have 30, I think this is not actually 30 parts per billion isoprene. This is not labeled correctly. But we have some isoprene that's present. And we can see that there are now some absorption features that are showing up. In terms of the light that's reaching the detector, we get less light at specific frequencies. And that's the characteristic of absorption spectroscopy. Now, to express this as absorbance, we calculate negative log of P over P naught of the transmission, and that gives us this spectrum here. So we can see that absorbance increases because of this negative sign. It increases when the amount of light decreases in comparison to what was measured originally. And so we can see for isoprene, we get these clear peaks in our absorption. And these are what we use to monitor the amount of isoprene that's present in our samples. So I just thought this is kind of cool to show you actual real-world data that was taken by students downstairs in my lab. And so this is how we measure absorbance.